Good afternoon, everyone, and thank you for being here. My name is Zack, and I'm a senior scientist representing Animatrix, a contract research organization based in Finland that specializes in gut microbiology across animal species. I have to preface that while I have done a lot of work with the partners in the agriculture and livestock industries, I'm still relatively new to the zoo and the wildlife industry. So at any point of time in time that if I make any references or that show my lack of knowledge, for example, zoo operations, please feel free to enlighten me. Right? Okay, so in the next 10 minutes, I would like to share on how non-invasive gut health monitoring can support long-term welfare, adaptation, and breeding success in avian species. It's an evolving concept based on emerging evidence, something that we are looking to collaborate together with any interested parties across the zoo, especially the zoo and avian communities. So after this presentation, if I've managed to convince you on how promising and exciting this approach is, please get in touch with me, find me for a chat. I'll be more than happy to share the final details. The microbiome is an ecosystem, a community of bacteria and other microorganisms living in the same space. The microbiome is dynamic and complex, with immigration, emigration, fragile coexistence of microbes within the same space, and animals typically thrive when the whole microbiome is in balance. In other words, a healthy gut depends on two things: balance and diversity. This image shows the beautiful three-dimensional structure of microbial communities. This is of an oral microbiome, but illustrates very clearly and similarly the complexities that we also see in the gut. There are many factors that influence the gut health, and I'm sure zoos put in tremendous effort to optimize and control various parameters, but I believe, and please correct me if I'm wrong, that typically only diet and environment, and perhaps animal behavior to a certain extent, can be reliably managed. But uncertainties and many uncertainties and occasionally surprises may still remain, which is why I have emphasized that it is prudent, therefore, to regularly monitor for abnormalities, which I'm sure many of organizations do. But to what extent? Different types of birds, like frugivores, granivores, and carnivores, each have their own version of a healthy microbiome. They don't share one ideal pattern. Balance simply means their core bacterial abundance levels stay stable over time, and without sudden spikes in any of the stress-linked groups. What matters is functional stability for that species, be it protein fermentation for raptors like vultures, or fiber fermentations for parrots. And this balance may look different across species, but the principle is the same: a stable, resilient microbiome that matches their biology. You may ask, why does gut health matter? When the microbial balance is disrupted, or in other words, dysbiosis, we often see increased risk in diseases, inflammation, poor nutrient absorption, and higher sensitivity to stress. When you see all this, well, that means I want to say this: gut health is even more critical in zoos and aviaries, where many species are exotic, fastidious, and there for a long time, and also exposed to the public, which introduces all kinds of stresses like foreign sounds and smells. While I understand that zoos also put in a lot of effort in curating diets for each animal, I want to emphasize that gut health monitoring is, in my eyes, almost obvious in complementing the welfare of the animals. Nature itself knows the importance of a good microbiome, and in fact, we have observed that many birds evolve to partake in something known as coprophagy, in other words, consumption of feces. Earlier, this behavior was thought to be mainly nutritional, but recent literature suggests that birds acquire this behavior to get beneficial microbes through coprophagy, helping to stabilize and diversify their gut microbiota. Using the Japanese rock ptarmigan as an example, Yeah. Using the Japanese rock ptarmigan as example, the chicks develop this natural behavior to consume fecal feces from parents to establish gut microbiome for healthy development. Such actions reinforce the fact that nature evolves strange behaviors to improving and supporting gut health. A team of scientists in 2023 studied breeder hens and found that high egg-laying birds had more diverse microbiome activity. When microbiota from high egg-laying donors was transplanted into low egg-laying recipients, using this procedure called fecal microbiota transplantation, or FMT, researchers observed a few things. First, increased diversity in the microbiota, enrichment of beneficial microbes like Enterococcus and Lactobacillus, improved jejunal health, and overall ovarian function. So these changes were linked to the SIRT1 signaling pathway that shows the influence of the microbiome at the cellular level. I'm not trying to imply that you should do FMT in zoos. Of course, this needs to be carefully considered. But it highlights one important thing, that microbiota diversity can impact reproductive physiology. In another study, several groups of unvaccinated chickens were challenged intranasally with H9N2 avian influenza virus. Well, looking at the top image, infected control birds, infected control birds actually were not, there's no treatment, and they developed severe tracheal lesions, loss of cilia, there's mucosal hyperplasia, inflammation, and congestion. So you can see the edema in the submucosal area, and you can see some thickening as well. So there's severe inflammation. The thickening of mucosal layer is actually indicated by the yellow triangle, which is a bit small here, but it's just trying to show you the extent of the damage by the avian influenza. At the bottom, you can see that it's healthier. Well, these birds were actually given probiotics intranasally with this mixture bacillus plus lactobacillus. So they have much milder lesions and a healthier tracheal epithelium. Although this is a respiratory example, it illustrates a broader point that microbial modulation enhances tissue resilience and immune responses. Now that we have a better understanding of how important gut microbiome is, how exactly should we characterize the microbiome? Instead of just asking what individual bacteria are present, we can go one step further to study the functional clusters to understand what they are doing. We know that certain microbes maintain the gut barrier, keeping the intestinal lining strong to keep out the harmful bacteria from getting through. Some regulate inflammation by calming down the immune system so that it doesn't overreact to stress and minor challenges. And some microbes actually produce short-chain fatty acids like butyrate, and these compounds fuel gut cells, reduce inflammation, and support overall resilience. So therefore, instead of just asking which microbes are present, functional clusters tells us whether the important work that is inside the gut is being done well. What I'm trying to say is that bacterial markers can identify what's going wrong and guide us on what action to take. It is often too late when clinical signs manifest in zoo animals, which can be very costly. We want to prevent this, and by leveraging on bacterial indicators, we can get early warnings of diet issues, stress, inflammation, pathogens, and environmental problems before even the clinical signs manifest. Take for example that it's actually possible to predict early signs of dysbiosis by looking at 3 specific bacterial markers: Enterobacteriaceae, Pseudomonas, and Aeromonas, which can indicate stress, inflammation, and high susceptibility to diseases. This will signal to the vet or the zookeeper to perhaps maybe have to investigate, do a hydration check or a diet check, or any other measures to prevent the dysbiosis from exacerbating further. Well, let me walk you through an example of how such indicator panels could be used. Imagine that you are in charge of overseeing the eagle enclosure and overseeing the health. And as part of a routine health surveillance program, you take the eagle fecal samples and you test it for dysbiosis using an indicator panel. And one day out of the blue, you saw that, oh, there's a rise in the indicator levels. Perhaps there's no significant adverse events that had happened, at least in your memory, but the change in the trend would still prompt an investigation to find out the root cause. And perhaps during the investigation, you found that, oh, maybe one of the feed ingredients was found to be expired, maybe. But my point is, this early warning could have allowed you to rectify the issue before the animal gets visibly ill. Conversely, the panel could also be used to measure stress reactions to unusual events, like construction. Well, let's use this as an example that maybe the construction work is happening nearby, and perhaps you want to use the panel to check if your eagle is getting affected, and then you can decide whether you want to move your eagle to another holding area before the construction, until construction completes before you move it back, maybe. Another scenario could also be that you want to introduce a mating partner to the enclosure and want to monitor the stress levels of your eagle in a holistic manner to maybe improve the breeding chances. So measuring the individual bird's microbiome may also represent a new dimension for the breeding programs by identifying animals with a more resilient microbiome and a higher adaptive capacity. In a nutshell, bacterial markers can offer a quantifiable, objective way to understand fluctuations in the gut health even before clinical signs appear. As I've mentioned at the start that I am actively seeking to partner with zoos, avian and animal institutions. So in such a collaboration, this is what you might expect, and the process is very, very straightforward. First, we will provide you the collection kit, and the animals do not need to be trained to poop in a certain way or in a bucket or any way. You just simply need to use a collection kit that we provide, scoop 1 gram of the fecal sample, pop it into the collection vial, shake it, and send it to us. There is no need to use dry ice or -20 or -80 degrees Celsius fridge or storage conditions. And once we receive the sample, we will analyze them, provide you the gut health insights, data, or report, and we'll teach you how to interpret it. Everything is non-invasive, at least according to design. And we will also try to design it to fit easily into your routine zoo operations with minimal disruption. So how does your organization typically monitor avian health? If you're already collecting fecal samples, then it's fantastic, because why not incorporate gut health check into your routine? So you can gain more insights with the same samples. For zoos that rely on restraint or sedation for health checkups, this fecal-based method will allow an even more frequent monitoring in a safer, more gentle manner for both the keepers and the animals. Well, if you choose to partner with us, this is how the timeline will go, at least in my imagination, because we will still be looking for a partnership and we will always want to discuss more on this. So at the start, I will still propose to collect at least 20 to 30 healthy samples to establish a baseline. And once the baseline is established, we can begin to monitor and track individuals or groups over time. And at this point of time, you should start gaining insights on how diet, enrichment, environmental changes, or stress events affect the microbiome. So with continuous tracking, we move towards optimization, adjusting management practices to promote a more stable and resilient gut. To summarize this, I have 3 key takeaways for you. 1, a healthy gut is vital for long-term animal health. 2, non-invasive microbiome monitoring have a strong potential to improve nutrition and breeding strategies. 3, with enough data, we hope that the zoos and the aviaries can control the microbiome to optimize the animal welfare to their advantage and longevity. We have seen very positive results with our clients in the agriculture and livestock industry across Europe and US, and we firmly believe that this could really revolutionize the monitoring of the animal surveillance and the welfare planning in the zoos and aviaries as well. And with this, I've come to the end of my presentation. Thank you for listening to me, and I'll be happy to take any questions.
Thank you, Zack.